Okay, I think we'll give everybody just another minute here to log in, and then we'll get started. Okay, I think we'll go ahead and get started here. I don't want to take any time away from our presenters today. So first of all, welcome all of you to the third student e-poster session that the QED-C Workforce TAC is hosting. These have been all great sessions that we've had up to now. They've shown you the broad variety of research that's being done in the quantum area in different universities around the country. And we have another great lineup of speakers today. So I want to thank also, of course, all of the student presenters today. Really appreciate your taking the time to do this for us. I know that these are extraordinary times. There's a lot of stress that you're having to deal with, and including not being able to probably get in the lab and so forth for some time now. So again, we really appreciate your time and sharing your research with us. Even if it's something that you aspire to and haven't been able to complete recently, this will be a great opportunity, I think, for the QED-C community to network with you and hopefully follow up as time goes on if they would like to. So this is the agenda that we have for today. We have Mark Keppler from Texas A&M, Marissa and Yuanheng from Indiana University, Simon from UC Santa Barbara, and Ho-Lun from Virginia Tech. And so with that, I'm going to turn it over now to Mark. I'll stop sharing my screen and you can start sharing yours.
All right, everybody can hear me?
Yep, looks good.
So I'm Mark Keppler. I'm a PhD candidate in Professor Vladislav Yakovlev's laboratory at Texas A&M under the Department of Biomedical Engineering. So I'm going to talk to you about nitrogen vacancy sensors and what we're attempting to do in terms of bringing those into biomedical science applications. And there we go. All right. So as it is right now, we have many different voltage probes that we have access to. The gold standard, of course, is patch clamp. So patch clamp is a fantastic technique because we get amazing temporal resolution, great signal to noise, but the issues in terms of probe size means that you can't get lots of probes in a very small region of space, which limits your spatial resolution. You're not going to get something on the order of an image. And of course, because of the size of the probe, you're also not going to be able to attach to smaller objects like dendrites, axons. So you're mainly limited to what you can get in terms of the somal body in response. So we've gotten away from this over the past couple of years. We've seen a tremendous rise in the number of voltage sensitive probes that are available. And this gets us down to a molecular scale probe, which has a decent temporal response. They're getting better and better all the time as we find new dyes. And at the same time, we can attach these through functionalization to various proteins throughout the cell body with different wavelength, both excitation and emission criteria. So there's lots of different dynamic routines that we can go ahead and take advantage of. But these come with the limitation that they generate reactive oxygen species, they alter the membrane potential, they disrupt conduction velocities within the cell. So by having these molecules attached to the cells, we're fundamentally changing the system that we want to understand. And the idea is, well, how do we minimize probe interference while still getting something on par with a full resolution image? So the thought is that the nitrogen vacancy sensor has come around, and this is a fantastic dipole center that's found as a point defect within diamond. So we can see there's a vacancy in the center here, co-located near nitrogen in this tetrahedral configuration that we would find in the diamond lattice. And this particular sensor is sensitive to magnetic field responses. Not only is it sensitive to magnetic field responses, but it does so at room temperature, and which makes it fantastic for biological systems. We could take, say, one of these chemical vapor deposition chips made out of diamond, and we can implant nitrogen vacancies inside of the top layer near a sample that we would put on top, and we essentially have a lab on a chip model. We have a way of putting different magnetic analytes, functionalized to different groups, and looking for those analytes, or we can look for signals that are coming from cellular structures that are bound to the diamonds as well. So we might have a way to monitor cellular communication networks in vitro, but still there's a lot of applications for this that are emerging. There's a lot of interest in the lab on a chip models and developing things that we could potentially look at in terms of diffraction limited imaging. So using a CCD. We could use this as a test bed for pharmacological agents. Go ahead and look at the cellular communication and then disrupt that communication with pharmacology and see what the outcome would be. Also localizing paramagnetic particles, which has become of interest in late, especially with the gadolinium based contrast dyes and MRI imaging. We want to know where are those going. So in the bottom left image, we can see paramagnetic gadolinium bound to cells, showing a contrast with the cell membrane in the intensity map on top of a bulk NV sensor. And the idea here is, well, great. So if we have gadolinium inside of cells, how long does it take for that gadolinium to completely be removed from the body? Where does the leftover gadolinium bind? Does the leftover gadolinium cause signal changes within the environment? We can see that, we could potentially see that with nitrogen vacancy diamonds. And at the same time, we can look at the magnetic field within the environment, as we can see in the top right image, and not only get magnetic field intensities, but because of the spatial configuration of the nitrogen vacancy dipoles, we can actually get vector field maps out of this as well. So this is kind of the interest before we get into the theory behind it. As far as the theory goes, it's relatively straightforward in the basic readout techniques. So you can read these out using all optical techniques, or you can go ahead and modulate the microwave field. And it exists as a spin triplet. So we have a magnetic spin state 0 in the ground and a magnetic spin state ±1, which is really excellent because we could see Zeeman splitting in the ±1 state. So if we excite this with a 532 nanometer laser and look at the fluorescence, we notice that in the ±1 spin states, we have a 30% population drop out to a non-radiative state. And that transitions down to magnetic spin state 0. And this means that population conversion can be seen optically through use of a photodiode or a PMT. So what we can do is we could use a microwave frequency, and if we scan over the intensity, we'll find that at certain points that correspond to the band gap between magnetic spin state 0 and plus and minus 1, we have this drop in intensity. And because the two states experience Zeeman splitting under magnetic fields, we can look at the difference in the peaks in terms of frequency band, and we can correlate that directly to the magnetic field intensity. And using that, we can respond to transient magnetic fields, such as action potentials, as we're currently interested in, and see that as a perturbation in the Lorentzian. So the reason why we're interested in this is because of work that was done by Barry back in 2016. So this is one of those hot research items where he showed that we can measure the magnetic field in not only excised neurons, but also in live flatworm models. So he laid a neuron in orange here across a bulk NV sensor. And by doing that, this is the physical setup here. He was able to measure out a magnetic field signal. Now it's 1,650 averages, so it's not a one-shot process by any means. And it's also a triggered process in that he's getting a single snapshot. This is not a dynamic event. He's not showing the signal propagating along the axon, and he's not showing spatial variations within the system. It also should be noted that this is done with bipolar electrodes, so it's probably a stronger stimulus than we would expect. And so the research interest that we're trying to get towards, or the direction that we want to pursue, is can we take what's here and work it towards dynamic models, be able to watch the propagation, be able to see spatial events. And that's where we're at. Unfortunately, because of COVID, I have been out of the lab. I haven't been in the lab for more than 2 weeks since March. So this research has effectively been on hold. But I'd like to thank my collaborators who are on standby. And do we have any questions?
Great. Thanks, Mark. So I will open it up. I don't see any questions yet in the chat box. If you do have a question, I should have mentioned this at the beginning, you can either enter your question into the chat box, the Q&A box, or you can raise your hand, the little blue hand there, and I will unmute you. So do we have any questions? I'll go ahead and ask a question to get started here. So I probably missed it, but in the beginning you were talking about the existing techniques and that they had, spatial resolution issues and also timing issues, the response times and so forth of the different set probes. How is this probe, are you expecting in terms of its response time going to compare to the types of responses that you're looking for in the cells? Are they clearly going to be fast enough, you think, to capture the transitions that you're looking for?
Yes. So the transition or the transient response of the NV center is, I want to say, on the order of it's sub-microsecond response. So we're not at all concerned that it won't be able to see it's about a 3 to 5 millisecond event for action potential.
Okay.
So with that, we should have enough temporal resolution that we can capture the event. And as you saw in Barry's example, he had a pretty good resolution in the captured event, even though it was triggered and from a single source.
Okay.
So an aggregate of the dipoles within the system.
Right. Okay, thanks. I did see a question that came in now in the Q&A. It was actually, I think, pretty much the same question I just asked. So I think you've answered that one. These diamond centers, by the way, are they the vacancy centers? I guess this goes back to the spatial resolution and so forth, but would you, in the sample that you're preparing to look at, would there likely be more than one vacancy within that area? And if so, how would you, separate out the individual responses?
Yes, so these are going to be aggregates of NV centers on the diamond, and that also gets into another issue of uniformity. So right now, as far as I'm aware, getting a chemical vapor deposition diamond which has a completely uniform I guess, implantation across the entire spatial constraints of the diamond is kind of, it's not clear cut yet. Not every diamond is the same. Some people have sports cars. Some people have you know the other thing, not
Right.
a sports car. So, and I think that there's going to be a period of time where that's going to remain the case. Like right now, I think the industry standard in NV is at Harvard University in the Walsworth lab. I believe that's correct. They're down to something like 15 femtoteslas in sensitivity. And it's, that is the best in the business as far as things go. And that was done, or their sensor was used in that John F. Berry paper.
Okay.
So they not only have the industry standard results, they also have the the best sensor that I have seen published yet.
Okay. Okay, great. Thanks. Okay, so are there any other questions?
Yeah, I have a question. Do you have to worry about, maybe it's a silly question, but do you have to worry about your microwaves and your laser, like cooking your biological samples?
Potentially, yes. I don't believe so though. I've seen some techniques that I'm interested in where they have needle probes that you can use that have deployable antennas at the tips, and they're cooled probes. So they have a lumen that you can flush with saline solution in order to pull heat out of the local environment. And as long as you have a strong enough magnetic field that you can pump your diamond adequately, you should be fine. So you might be able to get localized response. But for our purposes, so that's going to be something that you'd be worried about if you wanted to implant like nano diamonds or micro diamonds or something of that sort. In the event that what we're talking about a thin strip of tissue on top of a chemical vapor deposition diamond. I don't think it's going to be a problem at that point. We should be able to adequately cool the sample.
Okay, great. Well, thanks again, Mark. That was really good, and we look forward to the results when you can get back in the lab. So with that, we'll move on to our next presenters. Marissa and Yuanheng, you can share your screen.
Okay, hello everyone. My name is Yuanheng, and today our topic is characterization of radial two-dimensional ion crystals for quantum simulation. And I will spend the first 5 minutes to introduce some basic information of our research. Then my partner Marissa will lead the next 5 minutes talk. 40 years ago, it has been clear that simulating a quantum system is a very challenging problem. For example, if we have n spin half particles, then 2 to the n numbers of state must be stored in the memory. And the time evolution of the system is a 2 to the n by 2 to the n matrix. As n increases, that means the classical computers are not able to handle it. Thus, we need another way to solve this problem. An alternative approach suggested by Feldman is to have one controllable quantum system to simulate another. This idea is opinion because it would solve both the problem of storing quantum state and simulating its evolution without any exponential explosion. And quantum simulation would be very useful for advanced error problems in physics, chemistry, and biology. For us, quantum simulation is a powerful technique to study the behavior of interacting quantum spins. many body physics such as frustrated system, spin liquids, high temperature superconductivity. In all cases, however, experiments required us to generate a quantum simulator with two-dimensional interactions. And the simulator could be realized in several ways based on the trapped particles. And our lab used ytterbium-171 ions as for the quantum simulation. And the technology for trapped one-dimensional ion chain is sophisticated. We can trap from one single ion to ion chain with up to 250 ions on the cryogenical system. However, if we increase another dimension to 2 dimensions, there's still a lot of technology details can be exploited. And current efforts to build our two-dimensional ion systems have 2 ways, by penny trap or microfabricated arrows. However, ions in the penny trap must solve its permanent 10 to 100 kilohertz rotation problem, and dealing the snow coupling race in the microfabricated arrows is also another issue. So we want to do something directly, realize a two-dimensional crystal in the Paul trap. And this is the trap we're used to create our two-dimensional crystals. By applying DC and RF voltages on each rod, we'll create a potential saddle, which is a quadruple potential inside the trap. However, the trap's imperfections will increase ions micromotion, and ions are not easy to cool at this situation. So we add some compensation electrode and carefully choosing compensation voltages. This will eliminate parts of this effect. And this is the pictures of the ion chain from our rod trap. Ytterbium-171 ions are cooled to around 500 microkelvin and separated by 5 microns. And the ratio of the axial and the radial circular frequencies will determine the shape of a crystal. So carefully measure the circular frequencies of our trap is important, and prediction of all frequencies of ions will help us to generate laser and ion interactions. And we use the console to simulate the trap's potential and get the trap's circular frequency by pseudo-potential assumption. Then we change the value of DC voltages and keep other parameters fixed at the same time. They measure the axial and radial circular frequency by linear feed the frequency from experiment and simulation. I got these 2 graphs. The fact is that we are using the RF voltages to drive our trap. This will need a driver motion called micromotion. When due to this micromotion, there is no longer equilibrium positions for all ions in the trap. Since ions are compressed in a single plane at the trap center, so there's effectively no micromotion amplitude along the axial directions, but the radial micromotion will behave like harmonic oscillations. It can be written in these formulas, where R0 is the average ion position, and other terms are higher harmonic micromotion terms.
In a standard trap where you have ion chains, the radial frequency is much larger than your axial frequency. So to get the ions into the type of two-dimensional lattice that we want, we can increase our axial frequency just by increasing the voltage on our DC end caps. And then as we do that, it pushes the ions closer together, and all of our axial frequency modes, except for the center of mass mode, end up shifting downwards. And at some point, the very lowest mode crosses 0, which corresponds to what we call a structural phase transition. And that's what we're showing here, a few different structural phase transitions. So as alpha, that's our axial to radial frequency ratio increases. You can see them go through the series of structural phase transitions from a chain at the top to a two-dimensional zigzag phase, back into the three-dimensional helical phase, and then finally into a two-dimensional lattice in the radial plane. There's a lot of applications like entangling or encoding these ion qubits during simulations where it's really important to know the exact locations of ions inside of these particular crystal configurations. It's also really important to know the trapping conditions that you need in order to induce those configurations in the first place that you want to use. So the right side of this image here, it uses a 3D position solver code along with frequencies that are extracted from our trap in order to calculate what those ion positions and brightnesses should be. And then those calculated positions, they're in good agreement with fluorescence imaging of trapped ions that are actually from our lab, which is what's shown on the left. The 1D to zigzag transition is very well understood, but there's increased interest, including, of course, our own in our lab, related to the 3D to 2D phase transition and the stability region for two-dimensional Coulomb crystals. So the plot that I show here has experimental data points for alpha, which again is our axial to radial frequency ratio, at these different critical phase transitions in order to help find and map those stability boundaries. We've plotted this data against the pseudo potential result for the 3D to 2D transition, and then against a well-known power law for the 1D to zigzag transition. The way that we make these measurements is shown on the right. We do this by injecting RF into one of the end caps on our trap, and then scanning through a frequency range. And then when the injected frequency is on resonance with the ion's center of mass mode, in any of our three principal axis directions, the ion heats up and we see a dip in collected fluorescence. That's on the bottom here. It's accurate to within a kilohertz. And so the fact that this data seems to follow the pseudo-potential approximation is promising because it points to a small amount of micromotion and thus small micromotion destabilization in the regime that we're looking to use. And so these characterizations that we've been showing with our current trap, what we're basically hoping to show with those is that a two-dimensional trap in general is suitable for quantum simulation. So I also want to talk briefly about the types of two-dimensional simulations that we plan on doing in the future and how to accomplish that. So you might have noticed in the pictures that our two-dimensional crystal is not actually in the camera plane. And so right now we aren't able to image individual ions the way we need to find our correlation functions or extract witness operators or look at how they're interacting. So we built a second trap for this purpose, which is a blade style Paul trap instead of rod. It's built out of tungsten. And having the blades closer together in this trap also allows us to get higher axial frequency, so it's got a geometry that's more suited for two-dimensional work. We currently have this trap in a second vacuum chamber that's pumping down, and it will be ready to go as soon as we're finished with this current study. So once we can get these site-resolved measurements, that'll allow us to study real materials that exhibit geometric frustration, starting with the unit cell of a triangular lattice. It has a ground state with a 6-fold degeneracy, and there's actually an extra degree of entanglement, which is seen when you break a global symmetry. So that'll be interesting. And then another example is a square lattice with next nearest neighbor interactions. If you look at a square lattice with nearest neighbor interactions, that has a known ground state. But if you introduce these longer range interactions, then the lowest energy configuration is no longer obvious and the ground state phase diagram isn't actually known. So we can do this by creating an interaction Hamiltonian that I'm showing here at the top. We do this basically with a laser field. So we apply a spin-dependent optical force via what's called the Mulmer-Sorensen gate, and it couples our spin qubits to the normal modes of motion in the system. So if we define our coupling here, Jij, in terms of our normal mode eigenvectors and our Rabi frequencies from the laser, we can actually get an effective Ising model. We can also tune our interaction range, and we can vary our frequencies, phases, and beam amplitudes to create a whole variety of different two-dimensional Hamiltonians to have in our toolbox. And then finally, in the future, it could also be possible to tune the lattice geometry of our crystal by using lasers to shelve specific ions into auxiliary states, so energy levels that aren't actually participating in the qubit in the quantum simulation dynamics. The example structures here are the Kagame lattice, the hexagonal lattice, rectangular, and spin ladder. And so in the long term, what we're looking at is directly observing the effect of this lattice geometry on frustration, entanglement, and the presence of exotic phases. And then, of course, I want to thank everyone in the lab who's worked on this, and also you guys for listening.
Okay, thank you guys. That was really great. So do we have any questions? I don't see any yet in the chat box or the Q&A, but you can type them in there, or you can use the raised hand function, and I
Stop
will
sharing.
mute you.
So I can't see the questions.
Yeah, I think you have to stop sharing the screen in order to see the chat box and Q&A. Anyone have any questions? So I have one, and I think you probably answered it towards the end there, but I just missed, I think, a little bit of what you were saying. So the laser is going to be able to allow you to entangle next nearest neighbors and not just nearest neighbors. Is that correct?
That's right. It's a little more ambiguous than that because so if we have that specific lattice, then there's only one next nearest neighbor, and we can do that. But in general, what we can tune is the interaction range. And so we have different couplings for different ions depending on where they are in the lattice, and it falls off from the center as 1 over R to the laser detuning. But we can go from basically an infinite range up to 1 over like R to the third.
Okay, thanks. Yeah, I do see one question in the Q&A here from John Burke. It's he says, how do you scale this to a larger ion number or larger ion numbers?
Yeah, so the current trap that we have should allow us to have about 30 ions. We also have plans in the future for a cryogenic apparatus that will allow us to get down to colder temperatures and scale up to about 250.
Okay, thanks. Yeah, I was actually going to ask you about the temperature. I didn't, I missed that you were operating these at currently.
Yeah, these are currently at room temperature, and we use laser cooling. We can get them down to millikelvin temperatures with Doppler cooling, and then down really close to the n=0 harmonic state. Right now we're looking at around n=8 before sideband cooling, which is like a further method of cooling. And then for the cryostat system, that would be at about 4 Kelvin.
Okay, great. Okay, I think that, let's see, I'll give everybody another minute here. Any other questions? Let me see if anybody's raised their hand. No. Okay, well, thank you guys again. That was fantastic work. Looking forward to more results on that, and appreciate the presentation today.
Thank you.
Thank you.
Okay, so we'll move on now to Simon, if you want to share your screen.
Okay, hello. Can everyone hear me and see the screen?
Yep, looks good and sounds
Okay,
good.
perfect. Okay, so my name is Simon. I'm a PhD student in Anya JH's group at UC Santa Barbara. And today I'm going to be talking about how one can use Diamond as a platform for exploring many-body quantum systems, and in particular, how you can tune dimensionality in such a system using Diamond. Why is this? Okay. So just before I move on, I just want to make sure that I acknowledge the fact that this is really a collaboration between three different groups. So there's Anya JH, my PI, as well as Kunal Mukherjee and Norman Yao. Kunal Mukherjee is now at Stanford, and Norman Yao is at UC Berkeley. And of course, the rest of the group. So I'm going to be talking about the Nitrogen Vacancy Center in Diamond. And so we've already learned a little bit about that, but just to review, shown here is a diamond lattice. So the blue atoms are tetrahedrally bonded carbon. And how you get nitrogen vacancy center is you replace one of these carbons with a nitrogen and another one with a vacancy. And usually when people say NV center, what they really mean is the NV minus center, which is the NV with an extra electron. And so it turns out that this is a spin 1 system, or a spin triplet. And the really great thing about NV centers is that they are easily addressable as well as easily readable. And you do this all optically. So the ground state is this ms equals 0 spin state here. And it turns out that when you shine a green laser on the NV center when it's in this state, it fluoresces in the red. Now what you can do is use microwaves to pump the system up into one of these higher spin states. And if you do that, then the NV center doesn't fluoresce quite as much. And so that's how you can quite easily tell the difference between the two spin states. In addition to this, by simply shining the green laser on the NV for a while, you can pump it into the spin 0 state. And so as we saw earlier, NV centers are useful for a wide range of things, but one very nice thing is that they make very good sensors of the magnetic field. And that is because the two dark spin states are separated by a Zeeman splitting of 4μ0b. And so just by varying our RF and looking for when the NV center gets dark, you can tell the magnetic field. But for our purposes today, we're not focusing so much on the sensing, but rather using the NV as a spin qubit. And so despite there being 3 levels, what we're going to do is we're going to just take 2 levels. So let's take the ms equals 0 state and the ms equals -1 state as our 2 states on the Bloch sphere. Okay, so now the next defect that I'm going to be mentioning today quite a bit is called the P1 center. So again, we have this blue diamond lattice, and then we remove one of the carbons and replace it with a substitutional nitrogen. And it turns out, due to a Yuanheng distortion, that there are 4 different P1 orientations. But over here is the spin 1/2 level diagram for one particular orientation. And what ends up happening is you measure the B field along one of these 4 axes. And so you actually get, if you have a whole bunch of these, 4 different lines. And so what that means is that you can selectively couple or decouple the P1 centers by addressing individual transitions. And then another thing that you can do is, so shown here, this is a P1 center. And so here's the level diagram for an NV center over here, and then the P1 center is right here. And so if you tune the field such that the splitting between the P1 center is resonant with the transition in the NV center from ms equals 0 to ms equals -1, then what you'll get is resonant transfer of spin between your P1 centers and your NV centers. You'll get a flip-flopping. So if your NV is in spin up, it can transfer that spin to an adjacent P1 and change the orientation. So why someone might want to do this is related to our work with Norm Yao. So what we're doing is coupling the NV center to nearby P1 centers using microwaves. And so if they're tuned on resonance, what you could imagine doing is pumping your NV into spin up, waiting some time for that spin to swap with a nearby P1, and then repeating it. And if you just do this enough time, the spins are going to start to diffuse out from the NV center, creating a soft shell of spin polarized nitrogens surrounding this NV center. And so then after you've prepared this state, you could ask, how does the state evolve as it approaches equilibrium, which allows us to investigate some thermalization of quantum systems, which is an area that right now has remained kind of experimentally inaccessible for a long time and has only recently become something that people can actually experimentally test. And so that's why we're interested in growing these very dense ensembles of spin. So my role is in the materials side of things. So I'm actually a diamond grower at UCSB. So at UCSB, we have a plasma-enhanced CVD chamber, which grows isotopically purified diamond, shown here. So we have a hydrogen plasma, then we introduce methane, and methane becomes radicalized and then incorporates carbon into the step edges. But what we can also do, so picture over here is a diamond lattice. So imagine we're growing up, and then at some point we turn on the nitrogen, and then turn it off again. We'll engineer some thin doped layer of nitrogen spins. So this is how we get confinement in the Z direction. And shown here is a fluorescence map. So fluorescence, because the NV centers fluoresce, fluorescence can be a proxy for NV center density. And so we can see here that this is going deeper into the diamond. You can see that it's really confined close to the diamond surface. So here's the diamond surface. The bright spot is NVs, and it's really quite confined. But how we can verify this even further is by using a technique known as secondary ion mass spectrometry. And so how that works is you take a primary beam of ions, in this case cesium, and you bombard your diamond with this ion beam, which causes various other molecular constituents to fly off. So then you do mass spec on whatever comes off of this secondary ion beam that comes off of your sample. And so by doing that, because the primary ion beam kind of blows up bits of your sample, if you do this as a function of depth, like going deeper into the diamond, you get the elemental concentration as a function of depth. So shown over here is one such Sims plot. So this is showing the intensity of atoms in counts per second. The black is our isotopically purified carbon. So we see a depleted layer of C13 wherever we grow. And this is because C13 has a nuclear spin, so we want to get rid of that. Then in red, we have our nitrogen density. So this is nitrogen 15. We see a very sharp 2D layer. However, another way that we might begin to think about exploring dimensionality further was kind of inspired by this other Sims plot down here. So we see two peaks, a red peak, which corresponds to the same 2D intentional doped layer. But then we saw this large blue peak, which was a bit of a mystery for a while until we figured that there's actually, there's two kinds of growth going on. There's the step flow growth that I mentioned earlier, but then there's also these hillocks, which are a particular kind of growth defect. And what we found is that the nitrogen incorporation into these hillocks is orders of magnitudes higher than into the bulk, which then means that, of course, our spin density is going to be orders of magnitude higher. What's kind of interesting on top of this is that the dense nitrogen is quite confined. And so if we actually measure NV centers on one such sample, we see that, again, it's much brighter, and this is a log plot. So it's several orders of magnitude higher than the regions around it. So there really are quite a lot of nitrogen and quite a lot of NVs in these hillocks. And because they're kind of spatially small, one can think about this as like yet another way of changing dimensionality. Like if you had quite small hillocks, you know, if you had very small hillocks and then you introduce nitrogen for a short amount of time, you might get a very small point of dense spins. So now that we've shown that we can actually control dimensionality quite well, in the future, we're going to actually try and observe transitions between dimensionality. So looking at how a system goes from 3D to 2D and what changes in the physics there. And then eventually, how does dimensionality also affect the pre-thermalization of a quantum system? So that's it. So I'm happy to take any questions now.
Great, thanks, Simon. Yeah. So I hadn't seen information before about hillocks there. That's new to me. That sounds very interesting.
We're still exploring it, but yeah, it's a potentially promising direction. For sure.
Right. Okay, so open for questions now. So again, you can put it in the chat box, the Q&A box, or use the raised hand function. There'll be a little blue hand that pops up next to your name on the participants list, and I can call on you to ask your question verbally. I'm going to throw out a question that maybe might be unfair because I don't know that you have looked into it at all, but I know that some people are also exploring using silicon carbide instead of diamond for some of these vacancy centers. Have you looked at that at all? And if so, could you comment on, what you think the trade-offs might be with silicon carbide versus diamond?
Yeah, so I think the primary reason why we're using so there's many different kinds of color centers. There's color centers in hexagonal boron nitride and silicon carbide. There's many different color centers in diamond as well. But the reason why we choose, or why we've chosen NVs is because we have a lot of control over them, because they have had a lot of, they've been explored quite thoroughly in terms of their sort of basic physics. A lot is known about them, which makes them kind of ideal probes for other kinds of physics. So I would say the NV center is particularly attractive because of this.
Okay. All right. And let's see, there is a question here. What is the concentration of NVs that you were able to achieve?
This is something that we're still trying to get a handle on, but in the tens of ppm in the helix. So we can achieve tens of ppm outside of the helix normally. So within the helix, we're still working on actually measuring the precise concentration, but it's maybe 100, somewhere between like 10 and maybe 30 or 50 ppm in terms of NVs, possibly. Our nitrogen is probably a lot higher than that too. Right, here's another question. Magnetic sensitivity can be limited by NV minus interactions. Are the P1 interactions lower or higher than NV minus? If the interactions are lower, is there a way to use this mechanism to enhance sensitivity? So I would say for most of the stuff we're doing right now, it's more the nitrogen concentration that's relevant. But in terms of sensitivity, the P1 interactions, I would say if you're trying to build a sensor, you mostly want to have just as high NV concentration as possible and low P1 concentration. So the NV interaction is stronger than the P1 interaction, I believe. But if you have a much higher density of P1s than NVs, then of course your P1s are going to be more important.
Okay, thanks. And there's a follow-up question to the one previously about the concentration of NVs that you achieve. This one is, with this concentration, do you see a power law decay in the T1 times?
So this is a very good question. So that's actually something we're working out, is how does so actually we're looking at T2 times for power law stuff. For T1 times, don't know that we've looked at that, but for T2 times, we definitely see a strange power law decay, which is somewhere between what you'd expect for 2D and 1D. And we're still sort of piecing together why we see something which is in this strange intermediate regime.
Okay, great. Okay, well, thank you very much, Simon. That was really good. Let's go ahead now and move on to our last speaker, which is Hulun. If you want to share your screen.
Can everyone hear me?
Yes, looks good.
So my name is Holden. I'm a PhD student from Economics Group in Virginia Tech. So my talk is about QED-C. So it is an algorithm for constructing answers for variational quantum eigensolver. So first, we want to ask in this so-called NISQ error, what can we do with the current noisy intermediate scale quantum devices? So one of the options is to implement hybrid classical quantum algorithm. And on the other hand, like people are interested in correlated system, like molecular system. And one of the things they want to study is the ground state energy of them. And to do that, we can do digital quantum simulation. So one of the most promising algorithm for doing that is the variational quantum eigensolver. And basically it's based on the variational principle where you prepare the answer state on the quantum device and also measure the energy on the device. And then you optimize the energy on a classical computer, and then you keep updating the parameters until the energy converge to a low enough energy, and then you can approximate the ground state energy. So it turns out that the performance of the VQE is critically dependent on the choice of the answer. So the so-called ideal answers we want to find is that we can span the Hilbert space we want, which is where the ground state lives in. And we can do that with the minimal number of parameters and also a minimal circuit depth. So right now, there are two widely used answers. So one is the chemically derived UCC-SD. The other one is the hardware efficient ansatz. So both of them have their advantages and disadvantages. For example, the UCC-SD can span Hilbert space efficiently because it is derived from the physical science that it can span the Hilbert space efficiently. But if you want to implement on a quantum device, then it will result in a long circuit, and also you came across some throttle error. So for the hardware efficient ansatz, you can also span the Hilbert space if you have enough layers, but then the Hilbert space may not be efficiently explored, because whenever you want to implement an entangling gauge, you need to implement the whole block of entangling gauge, while you may not want all of them. So there is a previous work of our group, which is ADAPT. So it is an algorithm to iteratively construct an ansatz for VQE while using the VQE as a subroutine. So first of all, you need to define an operator pool, so where the ansatz is building from, and then you measure the gradient in the sense that to study how much energy can be changed if you add this operator in the ansatz. So this part can be done in parallel. And then if the gradient is big enough, then you select the operator with the largest gradient and then add it in the ansatz. So, and then you do the ordinary VQE to update the parameters. So you keep doing that until the gradient is small enough, then then call finish. You can have the adapt answers. So the good thing about this algorithm is that it can use a minimal number of parameters and operators to construct an answer for a given pool and a given Hamiltonian. So there is a comparison between the adapt and the UCSD. So we can see that the accuracy is even higher with a lower number of parameters. But there are also challenges for this algorithm because a minimal number of parameters and operators doesn't mean minimal circuit depth. So we found that the circuit depth really depends on the structure or how you define your operator pool. So let's take a look at the original fermionic pool of the adapt. So basically it's just single excitation and double excitation operator. But if you want to implement on like devices, then you need to do a Jordan-Wigner transformation or other kinds of transformation. So that will lead to a huge sum of Pauli strings. And if you want to implement an exponential of this operator, then it would require a lot of entangling gates, which we don't want. So we have the qubit adapt. So we replace the original fermionic pool with a qubit pool, which consists of individual Pauli strings. And these Pauli strings are not just random, all the Pauli strings we can have, but the Pauli string appear in the original fermionic pool so that we can ensure the completeness, but not that huge pool. So basically, these Pauli strings has a mass length of 4 and potentially give non-zero gradient. So here is a result of a classical simulation we done for 3 molecules. So we plot the energy error against the number of parameters, and the blue curve is the qubit adapt, while the red curve is the adapt. We see that the qubit adapt use more parameters to attain the same accuracy. But if we look at the number of C0, which is the circuit depth, we see that the qubit depth can greatly reduce the circuit depth even though we have more parameters. While the green curve is the transpile result of the original adapt circuit. So we have a trade-off between reducing the circuit depth with an increase in the parameters. But this trade-off is beneficial for the NISQ device because the quantum resources is more expensive. But on the other hand, we have an increase in the size of the pool because we are using the qubit pool. And remember that we need to do a measurement for all the operators for the gradients when we do the adapt iteration. And it means that we need to do a number of measurements proportional to the size of the operator pool. So in order to reduce the cost of measurement, we want to reduce the size of the operator pool. So we try to do that randomly first. So we keep reducing the qubit pool and see the performance, how it converges. And the original pool size is like 400 something. And you see that the performance is similar. It can converge like within the similar number of parameters until the 1/33 limit. Then we see there is a stop of the algorithm at some point. So we see that the qubit pool is overcomplete. While we can reduce the pool size, but we need to do it systematically, avoid the incompleteness. And then we show that there is a so-called minimal complete pool can be constructed. It has a size of 2n minus 2, where n is the number of qubits. and it can be constructed systematically. So in here, we show some classical simulation of 3-qubit, 4-qubit, and 5-qubit random Hamiltonians with random initial states. So we see that there are two minimal compute pools we form systematically can always converge, and also the other minimal compute pool we search by brute force, they can always converge, while the incomplete pool cannot. They always stop at some point, and yeah, and so we can see that the importance of the completeness of the pool. So the last slide is about the related work of adapt QAOA. So basically, it is a modification of the original quantum optimization algorithm QAOA circuit with the concept of adapt. So it can dramatically speed up the convergence for some masked problem. So in here, we show the masked result for two particular graph type, and the green curve here show adapt result for one kind of pool, and the orange curve is a adapt result for another pool, and the blue curve is the original QAOA. So we see a greatly speed up of the QAOA convergence. So in conclusion, we can say that the adapt can allow the Hamiltonian dictate its own ansatz with a minimal number of operators and parameters, while with using the qubit pool, a relatively shallow circuit can be found. And to minimize the measurement and the circuit preparation cost, the size of the operator pool can be reduced to 2n-2 with a systematic construction of those minimal complete pool. So this is the end of my presentation. Thank you for attention.
Okay, thank you very much, Olun. So the talk is open for questions now. I have one question. If you could go back to the previous slide right before your summary. Yeah, I guess I might have missed it here, but why do you get this dramatically faster conversions using this method?
So the exact reason is still not very clear to us, but we kind of related this behavior to the so-called shortcut to the adiabatic evolution. And it shows that the choice of the adapt can always bring some operator in that simulate the adiabatic evolution. And that may cause a dramatically speed up in here.
Okay. Okay. Yeah, that's very interesting. That's quite an improvement. Okay. Do we have any other questions? I don't see any hands raised, but let's see. Are there any in the chat box or Q&A if anyone has any questions? I have one more general question, I guess. When you're using these hybrid classical quantum computing systems, and you're trying to design an algorithm, an optimal algorithm, how do you go about optimizing what part of the problem to run on the quantum computer versus the classical computer? Is there some rule of thumb that you use in order to do that optimization, or it really varies with each problem?
I think it's a good question. So I don't think there is a really rule of thumb right now, but guess it varies with different kinds of problem also. And I think at this moment, people want to try like keep using more like classical resources other than quantum resources, because right now the quantum resources is still expensive.
Right. Yeah, I guess, yeah, certainly over time, I'm sure this will evolve. Okay, are there any other questions here for Ho-Loon? Okay, I don't see any other questions coming in, so thank you again, Ho-Loon. That was great. I want to thank all of the students again today for taking the time to present. As you see, we have a wide variety of research topics going on. We don't really try to make it broad. It just happens that way. So that's really great that, just by natural selection, so to speak, we have a wide range of research topics that we get every session. So thank you all again very much. This was great, and we look forward to those of you joining us again next month for another set of student presentations. And again, just one last message for the students. If all of you could send me a copy of your slides, we'd appreciate it. And you could include, if you don't have them now, your email address if you would like to be contacted. We'll share these with the QED-C members, and they can get back with you if they have further questions. So again, thank you all very much.